ভালো করে দিস, ল্যাবের নামটা উজ্জ্বল করিস, ল্যাবের
Anirban Sengupta. It's from University of Calcutta.
Thanks for giving me the opportunity to, thanks to the organizers to give me the opportunity to speak over in this platform. So topic of my presentation is basically the proteasomal degradation and autophagy are co-regulated by this key player, the SQSTM1 or P62, which in turn modulates this NRF2 KEAP1 pathway in the cerebral malarial infection. So I want to start my talk with the NIH statement that has been given the last day, the yesterday itself, the April 25, on the malarial, the status of the malaria, that there is considerable progress has been made in this field for the prevention and the control, but there is a decrease in the worldwide rate of new malaria cases and malaria deaths decreased by 41 and 60% respectively. But in the very second paragraph they mentioned yesterday itself that the new cases in 2015 is almost 212 millions and almost 429,000 deaths occurred in the single year, and with every 2 minutes there is a death of one child in Africa. So on this occasion, interestingly, yesterday was itself also the National DNA Day for the commemoration of the DNA structure discovery. So we have already discussed, the other speakers have already have shown the pro and anti-inflammatory cytokine balancing actually plays key role in the disease progression in cerebral malaria. So the pro-inflammatory cytokine, if increases, it enhances the death of the host organism. So from that point we have take the cue that whether there is any inbuilt defense mechanism against these pro-inflammatory cytokines, against this inflammation. So we found that autophagy is itself a cell-mediated process which is an inflammation-related defensive mechanism against diseases, and it has been reported in various journals also. So this autophagy regulates various cytokines, and in turn various cytokines also regulates the autophagy. So this is a complex phenomenon actually, and this autophagy plays immense role in adaptive, innate and other effector functions of the immune signaling. So we have taken the formalin fixed paraffin embedded section of the spleen section, the spleen tissue sections after post-infection, and the control, and we stained it with the acridine orange, and the monodensyl cadaverine, the MDC staining, shows the positive autophagic vacuole formation, and also the TEM pictures shows the double membrane vacuoles, which denotes the autophagic vacuoles. We have used a particular dye from Enzo, which is a catealophilic dye, is an amphiphilic tracer dye, which actually specifically recognizes the autophagic vacuoles when the autophagosomes and lysosomes are fused, and not the only lysosomes. NDC or other stains do actually stains other lysosomes or the other low pH vacuoles. So from that we came to know that the autophagy is in process in the system post infection. So when we check about the autophagic gene expression both in the mRNA transcription level or in the translational level, we find that the autophagic genes like Beclin-1, LC3B, etc. are upregulated. It's fine, but one interesting finding that we have seen that P62, one gene which should be downregulated if the autophagic progression and the autophagic degradation is completed, that gene is upregulated. So this is the first clue that we got, and we are puzzled at this point that what is the actual scenario, that why this P62 level is high even if the autophagic progression is happening. So we actually P62, this is a P62 molecule if you see, and this is the protein that is destined to be degraded by the in the autophagic vacuoles, when they are ubiquitinated, these P62 molecules take this ubiquitinated protein into the autophagic vacuoles where the lysosomes got fused, and then the degradation by autophagic mechanisms actually do happen, and in turn the P62 should get degraded and its level should get lowered, but we find the opposite picture. So now when we are studying the structure of the P62 gene, we find in the 3' end there is 3 important domains, that is LC3 binding domain, there is keep1 binding domain and ubiquitin binding domain, and the string analysis also shows us that this SQSTM1, this P62 basically interacts with this keep1, LC3B, etc. So from the recent reports we also find that P62 actually serves as a shuttling factor of few of the proteins like this TRK1, some polyubiquitinated proteins like tau, etc. These are just 2 references to the proteasome for the normal proteasomal degradation process. So we want to see that whether that P62 interacts with the RPT1 in the diseased condition more than that of the control condition, and that what we find in the immunoprecipitation with RPT1 and P62 and western blotting with P62. Next, when we studied the ubiquitination level by immunohistochemistry, we find that there is increases in the ubiquitination level, especially in the red pulp region of the spleen, and the ubiquitination by western blotting, and we have checked by the promega-luciferase assay kit of the proteasomal degradation that all 3 modes of degradation, proteasomal degradation, that is the trypsin-like, caspase-like and chymotrypsin-like, all 3 modes are increasing. That means the proteasomal degradation pathway is under process. So now we want to go into deep into the P62, that particular key molecule, that okay fine, they are increasing even if the autophagy is going on, and they are shuttling some of the proteins to the proteasome for the degradation. It's fine. Now is there any other role? For that we check the phosphorylation status of this particular protein. So the key important phosphorylation status are these three regions are the key points, like serine 403 when are phosphorylated, they are induce the positive, they signifies the positive induction of autophagy, and that is what we got in our samples. This is S349P. When they are phosphorylated, the interaction, it enhances the interaction between the P62 and keep1 molecule, and there is another T269P or S272P, but we haven't got any band from it. So we can conclude from this that this 2 process is undergoing from the phosphorylation data that we do have. So now this keep1 is an interesting molecule because in normal scenario the NRF2 keep1 pathway is the antioxidant pathway, antioxidant gene enzyme synthesizing pathway. This normal condition NRF2 keep1 is in the cytoplasm, and when there is an oxidative stress, this keep1 got detached from the NRF2, and NRF2 enters into the nucleus and ultimately helps in the act as a transcription factors for synthesizing the antioxidant enzymes. Interestingly, this P62 also has a binding domain for keep1, and this P62 also, when expressed in or when upregulated, can bind to the keep1, and the binding domain of the P62 with keep1 is same with that of NRF2 and keep1. So there is a competitive binding. So when this P62 level is high, in the same region is occupied by the P62 molecule, and keep1 cannot interact with NRF2, and NRF2 when free gets into the nucleus and ultimately upregulated the antioxidant genes. So in our data we also find that there is a considerable increase with the days post infection with the interaction between the P62 and keep1. And we have found that there is in normal scenario, there is with the increase in the infection, there is an increase in the oxidative stress level, ROS level increase. So today itself there is a presentation from Professor Utpal Tatu, who told that this oxidative stress might be responsible for the gametocytogenesis, for killing of the gametes of the malarial parasites. So whether this but this pathway, if gets activated, then what will happen? The antioxidant gene will be upregulated. Then it will ultimately resulting in downregulation of the ROS level, or the oxidative stress level will be reduced, and the gametocytogenesis might be prevented to some extent. So now we want to see that when NRF2 gets into the nucleus with that of the other antioxidative enzymes, it has the binding motif in the P62 promoter itself. And interestingly, within only 2.5 kb upstream of the gene, that is to we have found the 4 motifs where NRF2 can bind, 2 in the forward strand and the 2 in the reverse strand of the P62 gene. So what might be happening over here is that this P62 interacts with the KEAP1, frees the NRF2, and NRF2 get into the nucleus, and NRF2 binds into the promoter region of the P62 and upregulated the P62 itself, and hence it is in a loop, and hence this synthesizing and this loop will continue, and auto loop of synthesis of the P62 will continue. To find that whether actually this NRF2 binds into the P62 region or not, we have standardizes, we have done a chromatin immunoprecipitation, we have standardizes the DNA shearing process, and we have found that there is an increase in the P1 and the P3 promoter, this P1 and P3 promoter binding the post-infection with the control 4 and 8 days post-infection. So now we want to go into the details. We have dealt with it right now, that whether this differential binding, that when NRF2 will bind to P1 or when NRF2 will bind to P3, whether that determines something, whether that signifies something. And one of the things that might be signified by this differential binding of NRF2 to the P62 promoter might be that it might give rise to the different alteration of the transcript variant of the P62 gene. So this transcript, because many of the times it has been observed in other reports that the differential binding in the different promoter regions by the same transcription factor actually causes the transcript variation. So to check the transcript variation, so this is the full length P62 molecule. Again, this is the KEAP1 binding domain. So this we from the database we find there is a 2 major transcript variant, variant 1 and variant 2. And in the variant 1, a major portion is missing. This major portion is missing, and interestingly, this major portion which is missing in variant 2 actually takes out a part of the KEAP1 binding domain. So if the transcript 2 variant is present in our system in high level in comparison to the transcript 1, this transcript 2 P62 actually will not have the property or the ability to bind to the KEAP1, and it might break that loop. So we haven't studied that up to yet, but we have designed the primers in such a fashion so that we can identify that which transcript level is present in our system. It's a forward primer, it's a reverse primer, and if the 2 variants are present, then the length of this amplicon will vary, because P1 and P2 will bind over here in this case, and one will give a higher band and one will give a lower sized band, and that is what we are getting. And with the days post infection, the transcript 2 level is increasing in our system. So it might be one of the reasons that this particular transcript variant might produces the protein which lacks the capability or lacks the binding motif of KEAP1, and hence that loop that I have discussed in the last slide might get disrupted by this mechanism. So in nutshell, this is what is I am talking today, that SQM P62 actually interacts with the RPT1 and shuttles the protein for the proteasomal degradation, and it shuttles some of the proteins also for the autophagic degradation, and this SQSTM1 also interacts with the KEAP1 and frees the NRF2, and NRF2 bind to the P62 promoter itself, and the P62 synthesis is upregulated, and this loop is continuing, and this reactive oxygen species actually also does the same thing. In our future as our future plan, we will want to decipher these mechanistic details in much detailed manner, and also want to see that what is happening in the NF kappa beta pathway, because all things are pointing to the NF kappa beta pathway, because ROS high will increase, P62 itself increase the NF kappa beta 1. P62 actually interacts with several players of the NF kappa beta 1 itself directly. So we will, this is our future perspective. So finally I want to thank my supervisor, my department of Zoology, Calcutta University, the fellowship from UGC, and the project funding from the DBT, DST and BRNS. So I want to thank my colleagues and friends from the malaria biology group, Tarun, Keswani, Samrat, Sarkar and Saikat, Mukherjee. This is our whole group and this is our university campus. So thank you for your presence. Thank you.
Open for discussion. Any questions? If there is no question, then thank you very much for a nice presentation.
May I request the chairpersons to acknowledge Dr. Anirban Sengupta with a memento for his presentation. Thank you, Dr. Anirban. Thank you, chairpersons.
করেছিস কি হ্যাঁ ছয় সাথে করতে পারবি আরো কিছু
So my out to Dr. Sizu Susan.